Bismillahirrahmanirrahim. Starting with the name of Allah, the most beneficent and the most merciful. My name is Malaika and I am from the Department of PS Chemistry. Today in my presentation, I am going to explain an experimental procedure on how to dye fabric with different shades of wet, sulfur, reactive, and direct dyes. So for the apparatus, we'll need some eye protection, beakers, petri dishes, tongs, scissors, sample tubes, and some fabric. First of all, I'll explain some of the basics of the dyeing. So commonly dyeing process is carried out in two ways, either continuous or discontinuous or batch. So first of all, we'll have a look at the batch or discontinuous fabric dyeing. In this type of dyeing, a certain amount of material is brought under repeated contact with the dye liquor and then transported to carry out the subsequent process. The machines used in this process are winches or jets. Secondly, we have a semi-continuous fabric dyeing method. In this type of dyeing method, dye solution is applied by padding and then it's batched or stored in certain conditions for fixation. Basically, padding can have the advantage of high dye yield. Padded goods are usually batched, wrapped in plastic and left for some period of time for the dye to attach to the fiber or steamed to fix the dye quickly. Then we have continuous fabric dyeing process. As it is clear from its name that all the things will be happening at the same time in a continuous manner. So this type of process is aimed at higher production. Here, dry, undyed fabric enters at one end and dry colored fabrics leaves at the other end. That is why it is also called ready to dye. And a typical diagram is shown below. The machines used in batch process are winch, jigger, and jet. In semi-continuous method, the machines used are pad batch or pad roll. And in continuous dyeing method, the machines used are pad thermosol, pad dry, or pad steam. Now we'll have a look at some of the lab dyeing machines. First one is lab padder for dyeing. It is used for the resin finishing application of knitted and woven fabrics. The 400-meter wide padding mangle with a 125 millimeter diameter is a solid and compact table design made of stainless steel. Second one, we have curing machine for dyeing. Curing machine is used to fix printed or dyed fabric. The machine is already running in multiple textile industries in Pakistan and internationally. Now we'll have a look at the procedures through which different fabrics can be dyed with different dyes. So first of all, have a look at the dyeing mechanism with wet dyes. A typical recipe for wet dyeing is given, which can be accepted with alterations also. So first of all, we'll have a look at the mechanism for wet dyeing. So wet pigment in its pigment form or its insoluble form is it lacks affinity for cellulose. So it must be converted to some soluble leucoform. And this will be done by a reducing agent, which will be an alkali or sodium dithionite. Then after reduction, the soluble leucoform of the wet dye is applied onto the fabric. And after its application, it is again converted to the wet insoluble form. And this oxidation is done through either air or chemical process. And the last one, the most common, wash off or soaping process. So let me explain the general wet dyeing process. So the required way water for dyeing is taken in the dye bath and it is maintained at proper temperature. The reducing and dyeing temperatures vary from dye stuffs to dye stuff. The well-scoured wet yarn is entered in the dye bath and turned several times so that the affinity of the color may be uniform. The yarn is turned from time to time and care should be taken to keep the bath at required temperature and also to keep the yarn thoroughly immersed under the liquor. The exhaustion agents or the retarding agents are added to the dye bath depending upon the dye stuffs taken during the entire dyeing period. Excess quantities of both sodium hydroxide and sodium hydrosulfate should be present in the dye bath in order to keep the dye in the soluble form. At the end of the dyeing, the partly or completely exhausted dye bath must be kept in a distinctly reduced condition Otherwise, oxidation of the residual wetted dye takes place in the dye bath itself, leading to the apparatus turbidity. The dyed goods may then be removed from the dye bath and excess liquor, which contains the unexhausted wet dye, sodium hydroxide, sodium hydrosulfate, is removed as far as possible from the goods. The dyed goods are rinsed then with cold water and then subjected to an oxidation process, which is either an air oxidation or a chemical oxidation. The dye material is at the end treated in a hot soap solution or a synthetic detergent solution for 15 to 30 minutes. After the soaping treatment, the dyed goods should be rinsed thoroughly, and finally the dye material is dried. Now we'll have a look at some of the testing methods for wet dyes. So wet dyes are to be tested after the sulfur dye test has come negative. A 100 to 300 milligram dyed sample is placed in a 35 ml test tube, to which are added 2 to 3 ml of water and 0.5 to 1 ml of 10% caustic soda solution. After being brought to boil, a 10 to 20 milligram portion of sodium hydrosulfide is added, and boiling is continued for another 0.5 to 1 minute. The cotton is removed and then placed on a filter paper to oxidize. Re-dyeing of cotton to a shade differing only in strength from the original dyeing indicates that the wet colors are present. Now we'll talk about sulfur dyes. So a typical recipe of dyeing with sulfur dyes is shown. We'll head towards the dye mechanism of sulfur dyes. So like the wet dyes, sulfur dyes also lack solubility or affinity towards cellulose in their pigment form. So they also have to be converted into their leuco or soluble forms. And here the reducing agent used would be sodium sulfide. After that conversion, the soluble leuco form, which is dispersed or diffused onto the fabric, it is then again converted to its insoluble leuco form. Talking about some of the testing methods for sulfur dyes. So a 100 to 300 milligram of dyed sample is placed in a 35 ml test tube, and it are added 2 to 3 ml of water, 1 to 2 ml of 10% sodium carbonate solution, and 200 to 400 milligram of sodium sulfide chips. The mixture is raised to a boil and boiled for 1 to 2 minutes. The sample is removed, and to the test tube are added 25 to 30 milligram of white cotton and 10 to 20 milligram of common salt. After boiling for 1 to 2 minutes, the cotton sample is removed and placed on filter paper and allowed to reoxidize. Under these conditions, sulfur dyes re-dye cotton in a shade which differs from the original only in strength. That's all about sulfur dyes. Now we'll head towards reactive dyes. So general recipe, which can also be stated with alternations, is given below. We'll head towards its dyeing method. So its dyeing method is very simple, comprises of only 3 steps. First one is dye absorption. In this step, fiber is immersed in a dye liquor and electrolyte is added to it. Electrolyte here used would be NaCl. This electrolyte will basically neutralize the negative charge formed in the fiber surface and puts extra energy to increase dye absorption. So when the textile material is introduced to the dye liquor, the dye is exhausted along the fiber. Second step is fixation. As you can see in the figure below, that fixation means simply that the reaction of the reactive group of the dye will be held with the terminal OH or NH2 group of the fiber, and thus it will form the covalent bond with the fiber. This is a very important phase which is controlled by maintaining proper pH by adding alkali. The alkali used for this purpose depends upon the brand of the dye and the dyeing temperature. Generally, caustic soda, soda ash, or sodium bicarbonate is used as alkali depending upon the reactivity of the dye. This creates proper pH in the dye bath and do as the dye fixing agent. The third and the last step is wash off. A good wash must be applied to the material to remove extra and unfixed dyes from material surface. This is necessary for level dyeing and good wash fastness. It is done by a series of hot wash, cold wash, and soap solution wash. Talking about some of the testing methods through which we can test the reactive dyes. So its testing method is very simple. We'll just have to boil and water the fabric with a temperature more than 60 degrees in sodium hydrosulfide and caustic soda as in wet dyes. First, the color will come out and then that color will decolorize. Last but not the least, we have direct dye. So a general recipe of direct dye is shown below. Now we'll head towards this dye mechanism. First of all, we'll have to prepare the dye bath as obviously, and then we have to, and then we'll have a look at the dyeing process. So the dye bath we just prepared is set with the required volume of the stock solution of the dye, 0.5 to 1% soda ash and water to make the dye desired molecular liquor ratio. Then the processed material is entered in the dye bath at 40 degrees C and dyeing is carried out for 15 to 20 minutes. The required amount of common salt is added to in even number of installments at intervals of 10 to 15 minutes. The temperature of the dye bath is slowly raised to boil and is continued as this temperature for the period of 45 to 60 minutes and then it is cooled for 15 to 20 minutes for better exhaustion. The goods are then removed from dye solution, squeezed and dried. At last, we'll speak about its testing methods. So 100 to 300 milligram portion of the dyed sample is placed in a 35 ml test tube. 5 to 10 ml of water and 0.5 to 1 ml of concentrated ammonia are added and the mixture is boiled in order to bleed off a sufficient amount of dye for re-dyeing a piece of white cotton cloth. When a sufficient amount of dye has bled from the sample is removed. The white cloth weighing 10 to 30 milligram is placed in the test tube. 5 to 30 milligram of common salt is added. After boiling gently for 40 to 80 seconds and cooling to room temperature, the cotton is removed, rinsed and examined. So re-dyeing on cotton in an ammoniacal solution in the presence of salt to shade and strength comparable to the shade and strength of the original sample is direct evidence of direct dye. That's all from my side. Thank you.